Okay, let's start here. Okay, so we are going to go over the final chapter in the laboratory section of your guys' book. So this is for microbiology and immunology. So this is going to go over all the different things that aren't, like when we do the blood test, this is all the other things like when we do culture tubes or when we do a strep test or stool studies. All of those other little things that don't contain blood is what microbiology is, which, and also is, the study of microorganisms. But only 1% of microorganisms are known pathogens in the in the body system. Usually they are survived in the intestines. So they will induce an immune response in our bodies to produce molecules that'll come in our defense. Also, like the white blood cells. So if we get any bacteria or microorganisms, your white blood cells also go to fight off that infection. But microbiologists, they are identifying organisms that are responsible for a certain disease. So, but again, they're only 1%, or there's only 1% of microorganisms that are known pathogens. Does that make sense what that means? Okay, perfect. So yeah, so we'll be, helping to prevent the different infections with, microorganisms and everything. So, but mostly, basically, we're going to go over first in this first section here. Sorry, let me start that sentence over. In this first sentence here, we're going to go over essentially like what microbiology is and the different types of bacteria and how they're identified. And then on Thursday, we'll go into more specific tests that are done through microbiology. So right now, we're just going to mostly go over those different types of bacteria, what they kind of look like underneath a microscope, and also how to prevent yourself or what protective equipment you need to wear when dealing with these different types of specimens. So the different classifications of microorganisms. So you have bacteria, funguses, and protozoa. Viruses are not alive, so they're not technically considered microorganisms because they don't stay alive on their own. They need a host to attach to. So basically, they need to have like a protective shell. So like basically in what they're saying is must be cultured in a fertilized egg or tissue culture to survive. So again, because a virus has to have some type of host to survive, so it has to have consist of either of RNA or DNA covered by a protein shell. Otherwise, they can't produce or survive on their own. But once they have a host, they can multiply and then survive. So does that make sense why like viruses wouldn't be considered a microorganism? Okay, cool. That is still a test that we do in like microbiology, but it's different because again, microorganisms can survive and multiply on their own without a host. So okay, so the way that they name microorganisms is they use a binomial which this part's not gonna make sense, but basically what this is they're characterized by their shape, how they stain under the different stains they use to identify the bacteria, and their shapes. So basically, we'll get into that where I'll show you the different shapes of the bacteria and everything. But bacteria are single-celled prokaryotype organisms that reproduce by binary fusion. Identification of the bacteria begins with their observation of their morphology, so how they kind of develop. So bacteria, again, they're separated into different classifications based on their characteristics, their shape, and then the environment in which they survive and also thrive. So some bacterial infections may need a warm, moist environment, while other bacterial infections that are caused by a certain microorganism need something dry to survive and reproduce. So what they'll do when they get a culture, which can be something from like, say, your throat or a wound, or I mean, from all sorts of different places that you can get a culture from. Usually it's just a superficial type of culture, meaning it's not going to be, it's going to be more, how do I explain this exactly? Because you do still get cultures for like stool cultures and stuff too, but it's going to, it's as far as what I'm trying to say, like with the superficial part of it, is it's not going to be too much internal tissue. I'm not saying this right, I'm sorry. I'm not quite sure how to explain that, but basically, when you get the culture that you're going to then have to grow the bacteria, because what you're trying to do with microbiology is we're trying to make that bacteria grow. So again, once we get the, this is down in the laboratory, once you get the sample, they are going to put what they collected, they're going to smear it onto it's a round looking little container that they have stained and then put into kind of a warm area, and they kind of put it into different types of temperatures and conditions so they can see if that bacteria grows or multiplies, or if it does not, if it dies off. So, okay, sweet. So yeah, so there's 3 different types of ways that you can classify them based on their staining. So there's gram-positive cells, gram-negative cells, and acid-fast cells. So, that's weird. So on the next slide, I'll show you guys the bacteria shapes. But pathogenic bacteria assume 3 different shapes. You have brown bacteria, which are called cocci, rod-shaped bacteria, which are called bacilli, and spiral-shaped called spirali. Which, so that is kind of easier to remember, you know, if it's a spiral shape, then it's spirali, bacteria, bacilli, and then round bacteria, again, is cocci. So certain arrangements are also seen within different bacteria. So what you want to make sure to review is your 49.3 in your textbook. And then this is what, underneath a microscope, what these look like. So they stain the bacteria, and then that's what helps it to stand out. So as you can see, you have these ones that have kind of clear in the middle, and then have this outer layer that's red, and then you also have these pieces here that are blue and clumped together. So on this side, A is red blood cells, which is a gram-positive cocci. So you can see these are round-shaped, so those are why it's called the cocci. So over here in B is red blood cells with a gram-negative bacilli. So you can see these different shapes over here, how they're kind of different, and then the color of them as well. And the characteristics are a little bit different. So again, if we look at this, so the rod-shaped bacteria, the bacilli, when you're looking at this over here, this is what they mean by the rod-shaped bacteria, these little guys. So then underneath this picture, but I know this is kind of hard to see, but this is another example of the bacilli. So this is a smaller picture, but you can see this is the blue dye that's around it, but then in red, which is what shows us the type or the shape of the bacteria. So this is another example of bacilli, so because it's rod shaped. Okay, so bacteria and their oxygen requirements. So bacteria are also classified according to their oxygen requirements, so how they thrive and survive with or without oxygen. So those who do not require oxygen are called aerobes, or I'm sorry, those that require oxygen, sorry about that, are called aerobes, and those who die in the presence of oxygen are anaerobes. So just kind of like when we were going over the different types of anaerobic and aerobic culture tubes and everything. So aerobes or an aerobic type of bacteria would need oxygen to survive. So usually those bacteria are going to be things that are going to be like on the surface of the skin because they have, oh, sorry. So those are going to be the types of bacteria that'll mostly be on the surface of the skin because they need the presence of oxygen, where an anaerobic type of bacteria could be something like gastrointestinally. So, and then another type of bacteria that would need would be considered an aerobe would be something like tuberculosis, because it survives in the lungs and it thrives off that oxygen. Without that oxygen, then the tuberculosis bacteria would die off. So, is that making sense so far, Tahila? Perfect. Okay. So bacteria are also classified according to their physical structures. So flagella are long, thin structures that aid in propulsion or propulsion. Not saying that right. I'm sorry. Capsules are thick, gelatinous coats surrounded by the cell wall, and then endospores are intracellular structures that allow the cell to remain viable when environmental conditions are not favorable. So again, that was that slide that we were kind of looking at before that had those different shapes of the bacteria and the structures of them. So basically, bacteria, the way that it's broken down or classified again is based on their shape and also the environment in which they survive. So those are going to be key factors into identifying the bacteria, and then also recognizing if it, when you use the different stains, if the bacteria survives under gram-negative or gram-positive. I saw a question pop up. Prions?
Prions.
I'm not. Where?
What are prions?
I'm not sure. What?
Okay, so a prion is a bacteria that it causes bovine encephalopathy, Crutchfield-Jacob. There's another one, but basically it makes holes in the brain, and it is transmitted through nerve tissue of somebody who's infected.
Oh.
So by eating a cow that has it, or when you go and you have a root canal, and if the instruments are not sterilized properly, or any kind of nerve surgery where there's been other nerve surgery. That makes sense.
Oh.
Sorry, didn't mean to be the teacher for a second. Sorry.
No, that's okay. I thought it was something that you saw on this slide, and I was like, did I miss something?
No, I was just wondering if you knew what prions were.
I did not.
Are actually, I want to say, I think there's actually their own classification of bacteria. They're not any of these, but they are a bacteria.
Yeah, I did not know that. But that's definitely interesting to know. Okay. So some common diseases that are caused by bacteria are tuberculosis, UTIs, and I'm not sure what this is. I've not seen that before. Um, and
Legionnaires?
tetanus. I'm sorry?
Legionnaire's disease?
Yeah, I've not heard that before.
So Legionnaire's disease is caused by biofilm that is found in water lines, that is found in air conditioners, things like that. And it's a lung disease, and it's very common in like big hotels, things like that. But the most common place is in a dental practice.
Hmm. Hmm. Interesting. Yeah. But yeah, these diseases are classified too based on so once a patient tuberculosis would be is something pretty rare, but it is again something that survives in the lungs. But the the diseases, what am I trying to say? Sorry, I got all mixed up. So each of these diseases are basically based on the type of bacteria that are based on the bacteria that's affecting them. So if the bacteria in the lungs were caused by like the I'm sorry, I got mixed up. um, but basically, like tuberculosis is caused by a certain type of bacteria, and again, it's classified on how it survives in those environments. So, and also how like the route of transmission. Okay, so some unusual pathogenic bacteria that you might find. So would be chlamydia, mycoplasmas, and rickettsia, which are thin or which are tiny, unusual bacteria that fall between the size ranges of a virus and typical pathogenic bacteria. So again, do you remember why viruses are not technically considered microorganisms to human? Yeah, so viruses, they need a host in order to thrive and multiply, where certain bacteria, they can survive in certain conditions, but they don't necessarily need a host. So, yeah. So rickettsia are gram-negative, so they're transmitted by, so basically like if it was a tick, that would be what would spread rickettsia. Chlamydia requires a host cell for growth, so they're considered, or they were once considered viruses because they need to have a host in order to continue to grow. And then mycoplasmas have no EG. I don't remember, I'm trying to remember what that stands for exactly. In the cell wall, but are not, they're not necessarily parasites. So, so rickettsia and chlamydia, they don't grow in artificial media in the lab. So tissue culture and serological testing is required so we can identify them. Because they're not going to be ones that you can put on those slides and grow like you can other bacteria. Okay, so pathogenic funguses. So mycology is what the study of the funguses are, which are eukaryocytes that include unicellular leaves and multicellular molds. They're transmitted by direct contact with an infected person, prolonged exposure to a moist environment, and inhalation of contaminated dust or soil. Diagnosis is usually based on culturing or microscopic observation of samples. So fungal infections can be superficial. So things like athlete's foot, or a lot of times what's really common, and it's most common in the elderly because they are those who usually are on not necessarily bed rest, but basically are in the bed a lot. So in certain folds of the skin, or funguses can start to grow, or yeast will start to grow. Um, so it's usually a pretty superficial type of infection, but they're usually pretty easily treated as well, though. So, All right, pathogenic parasites and protozoa. So parasites harm its host while the parasite thrives. So there's different types of those parasites. Um, and usually those are identified through stool samples. So something like if a patient went to another country, sometimes certain water, or even too, if they like say they were out in the woods hiking and then they drank from a stream, sometimes those types of things can cause someone to develop parasites, which we'll usually find in their stools. So once they do the stool sample, we'll be able to identify specifically what type of parasite it is that's causing their infection. So those, you would send the patient home with a kit, which I think we went over a little bit on those different types of containers with the preservatives of what you would need for those. But we would send the patient home to collect those stool samples. And then once they come back, then we would, the lab would, if you were working down in the lab, we would then put them on a wet mount so we can see what type of bacteria it is. So we can look and see if they are caused by worms or if it is a single-celled protozoa. All right, I had a picture here for this. I think it's actually on over here. So let's look at what those worms look like. Can you still see my screen? Good. Okay. So this is what it would look like if we were looking underneath the microscope and then we were to find these types of parasites or this type of worm in a stool culture. So usually you can't see them to the naked eye. That's why you need to look at them under a microscope. So usually a patient will come in with certain symptoms of either diarrhea or upset stomach, frequently having to have a bowel movement. So those different types of symptoms can trigger us to then do a stool test. So, all right. So they're the different types of, helminths worms. So they live on or within another living organism and nourish themselves at the expense of the host organism. So this is, have you ever heard of like a tapeworm? Oh, okay, gotcha. So a tapeworm would be something that basically it's, okay, good. So it's surviving off of the host itself. So it's not going to, when it says like it's going to nourish themselves at the expense of the host. So what that basically means is like when the person is eating and gaining nutrients, the worm basically is going to absorb that nutrients, and then even though they need the host to still survive, they will still basically steal all of the resources from the host to make itself survive, rather than allowing its host to thrive. So it's more worried about itself thriving than it is the host thriving. So again, we do need to look underneath the microscope so we can identify them. And then stool specimens commonly examined for parastatic protozoas and helminths. So it's looking for those different forms here. So usually that test we would order would be called the ova and parasite test. I saw a question. Oh, I hope you don't have a tapeworm. So, okay, so viruses. So again, viruses are not considered a microorganism, but again, we still look at them under microbiology. But viruses consist of a core of either RNA or DNA that has to be covered by a protein shell. So again, when we are doing this in microbiology, we do have to make sure it's attached to some type of host that has a protein shell, which is usually in fertilized eggs or in a tissue culture. So Instead of culturing a specimen for a virus, sometimes we can do a blood sample or other bodily fluid that we can test for a specific antibody related to that suspected viral infection, which is usually one of the most common ways to test for viruses. Is they are testing to see if it responds to certain antibodies. All right, so specimen collecting for microbiology testing. So we always use a sterile collecting container. So that could be between, if it was like we were doing a culture on a wound, you would be then putting the, usually it comes in a kit where you have the container and then the swab together. So then you would put, once you take the culture, you put that swab into that sterile device, or maybe you have a sterile cup. But you always want to use a sterile collecting method and transport devices. And then we want to make sure that we prevent contaminating the patient any further, but also we want to make sure that they don't contaminate anything around where they are at as well. So once we're done, we do have to make sure that we really clean that room, so which we usually will use in those tubs, those purple wipes that we have. That's what we usually use to then clean the area, because we don't want anyone else to pick up any bacteria from that patient as well. Because again, that bacteria can last on the surface because it doesn't necessarily need a host in order for it to survive. So, but you want to make sure, depending on the devices that you're using to transport these or the containers, so you want to make sure you follow the instructions carefully. So is it something that has to be at room temperature, or does it need to be refrigerated? Or how soon does this need to be tested? Like once the patient then say they do the stool sample, how soon do they need to bring it back to the lab in order for it to still be a good sample? And if we were going to do certain specimen collections, so you want to cleanse the area to be sampled with an antiseptic, depending on what it is that you are collecting. So if it was going to be, say, an open wound on the skin, we wouldn't want to cleanse that area with the antiseptic, because then we're basically killing off any bacteria. So that really depends on what it is that you're collecting. But then you'll open the sterile containers when necessarily. So once you do the swab and everything, to then put it into the sterile container. But you never want to touch a sterile swab or collection device to a non-sterile surface. So once you open up that packaging that's together, so if you're going to do a wound culture where it has that package together of the swab and then the collection container, you don't want to take the swab out and put it on the counter, because then it can pick up any bacteria that's there. So we just want to make sure we take it from the packaging to the wound and then immediately into the container that we're going to be using. So, but as always, you want to make sure that you wear gloves. You don't have to necessarily wear a mask unless you are dealing, if we're going into a room where we're going to be dealing with a patient potentially with tuberculosis or with C. diff, which is a type of infection that, again, we would look through the stool to see. And those in hospitals, if you're going into a room that you have a patient that you're collecting a sample like that, or they're suspected to have one of those types of disorders or bacteria that is transmitted through air, they will have the precautions usually posted outside of the patient's room, as well as a cart that has all of the PPE in it. So that way you can put on the gown, your surgical mask, or if you need to have an N95 mask, goggles, and of course, as always, you always wear gloves. So, and yeah, of course, I don't know why it has to specify this, but even if you're like a fecal or urine specimen, you always want to wear gloves. So, but usually once you get down into the lab with the fecal culture, they will have it's protected kind of by like the plexiglass, but then it also has a ventilation system. So that way you are not going to potentially, even though you're going to be wearing a mask and goggles, in gloves and a gown, they will still use that vented hood so that way, because the bacteria that can be in a stool sample can be passed pretty easily. But they just want to make sure that you don't, it's not necessarily passed through air, but once it is exposed to air, it then can travel through air. I hope that makes sense what I mean by that. So, but yeah, do you have any questions, Tehila? Okay. So this was the last one I was going to do, actually. And then we're going to go more into, okay, yeah, because I know you said you took a microbiology class. But Thursday we'll go more into the specific tests that are done, or the most common tests that are done in microbiology. Okay, so collection for pinworms. So someone who is infested is usually an ingested egg through hand to mouth transfer. So sometimes, and this is something that is the most common way for this to be passed, unfortunately. But say someone goes to the bathroom and then they don't fully wash their hands. So if they have a parasite or one of the types of worms, they can then still be on their hands and say they go to them prepare food and aren't wearing gloves, and then someone goes and eats it. So that's kind of what it means, like hand to mouth type transfer. That can also be like if you touched an object that someone, again, they didn't thoroughly wash their hands maybe, and then you went to go like drink some water or you touched your face or something. So those eggs will hatch in the small intestines. And for kids, it's usually best to collect the sample late at night or early in the morning, because that's usually the time that they'll have a bowel movement. So, but yeah, so basically, again, so viruses are not technically considered a microorganism because they need to have a host to live. Only 1% of microorganisms are known pathogens. The different types or the classifications of microorganisms are bacteria, funguses, and protozoa. And the microorganisms and bacteria are based off of their shape, the way that they react to the stains that we will use, and also the environments that they survive in. All right. Do you have any? Did you have any questions or anything from like last week's lesson, Tehila? Okay, perfect. So yeah, again, on Thursday, we're going to go over more on the different types of testing that we'll do. And we'll go even further into collecting for those particular tests, like how to collect those samples and everything. Okay?